How mitochondria support T-cell proliferation and memory formation. New study. Mitochondria play a crucial role in T-cell growth and memory development. New center. In a groundbreaking study led by Dr. Navdeep Chandel, a prominent figure in the field of medicine, and the David W. Chandel, MD, Professor of Medicine in the Division of Pulmonary and Critical Care, researchers have unveiled how mitochondrial metabolism is essential for T-cell proliferation and plays a significant role in preventing T-cell exhaustion, particularly in the context of cancer and chronic infections. These findings, recently published in Nature Immunology, enhance our understanding of mitochondrial functions in T-cells and could pave the way for innovative immunotherapy approaches. Mitochondria are often referred to as the powerhouses of the cell, and their electron transport chain, ETC, is a series of protein complexes that facilitate electron transfer, ultimately leading to the production of ATP, the energy currency of the cell. Previous research from Chandel's lab had already established that the mitochondrial ETC is vital for the proliferation of CD8 T-cells, which are crucial immune cells that combat cancer and viral infections by recognizing specific antigens. However, the exact roles of the mitochondrial ETC in enhancing CD8 T-cell responses and proliferation had remained elusive until this study. In this investigation, the researchers engineered mice that lacked mitochondrial complex 3, one of the 4 complexes in the ETC responsible for electron transport and proton delivery, which are essential for ATP generation and the production of reactive oxygen species, ROS, that act as signaling molecules in cells. Observations from these genetically modified mice revealed that a malfunction in mitochondrial complex 3 led to reduced cellular respiration and a decrease in signaling molecules associated with ATP production. Consequently, CD8 T-cells with impaired mitochondrial complex 3 function exhibited diminished proliferation in response to viral infections. Surprisingly, these T-cells also experienced rapid exhaustion when stimulated with antigens, a phenomenon previously only noted under chronic antigen exposure. Moreover, the study highlighted that the dysfunction of mitochondrial complex 3 adversely affected the formation of memory in CD8 T-cells. This memory is crucial for the immune system's ability to remember past infections and respond more effectively to subsequent encounters with the same pathogen. Dr. Chandel explained, whenever you encounter a viral infection, your immune system mounts a vigorous response to eliminate it, leaving behind a few memory T-cells. These cells are essential because they can quickly proliferate if the same virus invades again, providing you with protection. To delve deeper into this issue, the researchers introduced an alternative oxidase, AOX, protein derived from C. intestinalis into the CD8 T-cells lacking mitochondrial complex 3. AOX can compensate for the loss of complex 3 without producing ROS. The results showed that while AOX effectively prevented T-cell exhaustion and helped restore cellular metabolism and proliferation, it did not facilitate memory formation, indicating that the generation of ROS is linked to the development of T-cell memory. Dr. Elizabeth Steinert, the lead author of the study and a research assistant professor of medicine in the Division of Pulmonary and Critical Care, noted, "We observed the expression of memory precursor markers in these cells, indicating that the issue is not merely an immediate death signal, but rather a failure to develop terminal memory cells that can persist." In summary, this research underscores the critical role of mitochondrial respiration in T-cell proliferation and memory formation. These insights could inform the creation of new therapeutic strategies aimed at targeting mitochondria within T-cells. Dr. Chandel emphasized, this research indicates that mitochondrial metabolism is key to preventing exhaustion, supporting proliferation, and generating memory. This positions mitochondria at the forefront of T-cell biology, suggesting that we should consider therapies that rejuvenate mitochondrial function. The study's co-authors include Beatriz Furtado Bruza, a graduate student in the Driscoll Graduate Program in Life Sciences, Rogan Grant, a Schmidt Science Fellow at Northwestern, Karthik Vasan, a former student in the Medical Scientist Training Program, Wegwo Kui, a professor of pathology in the Division of Experimental Pathology, and Samuel Weinberg, an assistant professor of pathology in the same division. This research was made possible through funding from the National Institute of Health grants R01 AI 148190 and 5P01 HL 154998, the Cancer Research Institute, and support from Schmidt Science Fellows in collaboration with the Rhodes Trust. Powering up T-cells, unlocking the potential of cancer immunotherapy. Powering up T-cells, a new path in cancer immunotherapy. Researchers have identified a novel approach to enhance the immune system's ability to combat cancer. By targeting a protein called ANT2, they reprogrammed T-cells to consume and generate energy more efficiently, effectively rewire their internal power supply. This shift elevates T-cells' activity, resilience, and effectiveness in attacking tumors. The findings open new possibilities for therapies that strengthen the body's own immune response, offering a more targeted and smarter approach to cancer treatment. medicalexpress.com/cancer therapy. Led by PhD student Omri Yosef and Prof. Michael Berger from the Faculty of Medicine at Hebrew University, alongside Dr. Magdalena Huber from Phillips University of Marburg and Dr. Eyal Gottlieb from the University of Texas MD Anderson Cancer Center. The international team discovered that fine-tuning immune cell metabolism significantly improves their capability to destroy cancerous cells. https medicalexpress.com/cancer. By disabling ANT2, we triggered a complete shift in how T-cells produce and utilize energy, explains Prof. Berger. This reprogramming made them significantly more effective at recognizing and killing cancer cells. https medicalexpress.com/cancer. In simpler terms, blocking this protein forces the immune cells to adapt their metabolism, transforming them into stronger, faster, and more aggressive cancer fighters. Published in Nature Communications. This study explores the mitochondria, the key metabolic hub of cells. By disrupting a specific energy pathway within T-cells, the researchers rewired their engines, creating a state of heightened readiness and potency. The altered T-cells showed greater stamina, faster replication, and sharper targeting of cancerous threats. Perhaps most importantly, the study demonstrates that this metabolic reprogramming can be achieved not only through genetic modifications, but also via drugs, opening the door to potential clinical applications. This discovery reflects a growing trend in cancer immunotherapy that focuses on upgrading the immune system's inner machinery rather than just guiding its function. While further research and trials are needed, by learning how to control the power source of our immune cells, we may unlock therapies that are both more natural and more effective. More information. Omri Yosef et al. Metabolic reprogramming driven by ANT2 deficiency augments T-cell function and anti-tumor immunity in mice. Nature Communications, 2025. DOI 10.138.S4146-025-593-3 https://doi.org/10.1038-593-0. Citation: Powering Up T-Cells, A New Path in Cancer Immunotherapy. Retrieved 29 July 2025 from https://medicalexpress.com/news2025/powering cells path cancer immunotherapy.html. This document is subject to copyright. For private study or research purposes, no part may be reproduced without written permission. The content is provided for informational purposes only.